damage, while treated group showed reduced swelling and improved joint condition. Maximum improvement was observed at higher doses. The mechanism can be explained through combined effect of phytochemicals. Phenolic and flavonoid compounds reduce oxidative stress by scavenging reactive oxygen species. This leads to decreased production of inflammatory mediators such as cytokines. As a result, inflammation is reduced and tissue healing is promoted. A strong relationship was observed between high phenolic and flavonoid contents, strong antioxidant activity, and significant anti-inflammatory effect. This confirms the biological activity of the plant is directly linked to its phytochemical composition. In conclusion, Gerbera jamsoni demonstrated significant antioxidant activity, strong anti-arthritic potential. The effects were dose-dependent with highest activity observed at 200 milligrams per kg. These findings suggest that this plant has promising potential as natural therapeutic agent for inflammatory diseases such as arthritis. Further studies are recommended to isolate active compounds, explore detailed molecular mechanism, conduct clinical trials for human application. Thank you for your attention. Allah Hafiz.
Malik.
Assalamualaikum. Everyone, I'm Dr. Sabah Zafar from the Department of Biochemistry and Biotechnology of the University of Multan. Today, I will present our work on predisposition
मैं क्या ले गया वा चाय बना दे यार
of FOXQ1 gene variants in pancreatic cancer from
ड्राइव में पता करना
future perspectives of our results. So, first is the introduction. Cancer develops when abnormal cells grow uncontrollably and invade healthy tissue. It progresses through several stages from localized disease to metastasis. Early detection remains the key challenges in the cancer. These are there I am showing the developmental stages of cancer: initiation, proportion, and spread. Pancreatic cancer basically begins when pancreatic cells become abnormal and form tumor. It can spread to nearby organs such as liver, lungs, lymph nodes. The most common type is pancreatic adenocarcinoma. The pancreatic adenocarcinoma is the most important type of the pancreatic cancer. But there I am showing the other four types of the pancreatic cancer as well. There is the anatomy of the pancreas. The pancreas is divided into head, body, and tail regions. Its anatomical location deep inside the abdomen makes pancreatic tumor difficult to detect in early stages. The pancreas has two major roles: digestion through enzyme secretion and blood sugar regulation through hormones like insulin and glucagon. Disease in this organ affect multiple body systems. Common symptoms of the pancreatic cancer include abdominal pain, jaundice, weight loss of appetite, fatigue, and digestive problem. Unfortunately, many symptoms appear only after disease progression. There are the risk factor of pancreatic cancer. We divided into main three categories. First one, the demographic risk factors includes age, sex, race, family history, diet, physiology, inactivity, coffee consumption, and alcohol use. While clinical risk factors include diabetes, chronic pancreatitis, infections, gallstones, metabolic syndrome. And pancreatic cancer is also influenced by the genes. So there are genetic mutations, random errors during DNA replication, and most important genes involved in the cancer are BRCA1 and BRCA2. There are some genetic factors that are associated with the pancreatic cancer. Several genes are associated with the pancreatic cancer, including BRCA1, BRCA2, TP53, ATOM, and others. Our interest is in the FOXQ1 gene, which may influence cancer growth and progression. FOXQ1 gene belongs to the Fock-head box family of transcription factors. It regulates cell growth, migration, epithelial mesenchymal transition, and tumor aggressiveness, making it an important cancer candidate gene. So there are the aims and objective of our research. Our objective was to investigate whether selected FOXQ1 shapes are associated with pancreatic cancer risk and whether these variants relate to the tumor features and biomarkers. Materials and methods. We obtained ethical approval, collected the blood samples and data collected from the patient with the consent of patients and controls. Then we stored the sample at 4 degrees centigrade. We extracted the DNA. We checked the quantity and quality of the DNA. Then we designed a primer by using the primer one tool. Then we do the genotyping for the selected SNPs and observe the results on the 2% dose gel electrophoresis. And then we do the statistical analysis, including the genetic factors, clinical factors, and demographic factors. So these are the PCR results of our selected SNPs. We of our selected SNPs, the gel image confirms successful amplification of SNP. We identified homozygous wild type, heterozygous, and homozygous variant genotypes. Similarly, the second gel demonstrates the SNP RS1124679 with the AAAG and GG genotypes, confirming the reliability of our laboratory methods.
हाँ जी
This is the
साहब
descriptive analysis of our research. We compared the demographic variables between controls and patients,
क्या हाल
including
है
age, BMI, and gender. Such variable helps
ये तो
understand population
हाँ जी
characteristics and the confounding factor. We also include the exercise, junk food, smoking, diabetes, and
मैं
present risk of
लेट हूँ
pancreatic
कॉन्फ्रेंस
cancer. Then we calculate the genotypic and allelic frequencies in cases and controls, and it
ये
also
साहब
shows
बोल
that our both SNPs are in the hardy with equilibrium. Then this is the this is the graphical representation of our genetic variants, and
अच्छा
it
ठीक
also shows the p-value for the association of SNPs with the pancreatic cancer. The our SNP RS950289 shows the
इसके जिम्मे रैन को भेज दो
significant
ये
association with the pancreatic cancer, and the third graph present the association of haplotypes within the pancreatic cancer, and it is shown here that the TCCG haplotype, TCAA haplotype, CCAG haplotype, and CCG
ठीक है
haplotype have significant association with the development of pancreatic cancer. Then there is the association of SNPs and haplotypes with the tumor size, height, and weight, and it shows that the red text shows that the affected individuals, if they have these SNPs, they have more. The tumor in this is more height and is more larger in size than the than the patient which have the wild type SNPs and haplotypes. Then there is the correlation of CN19, which is the cancer biomarker with the SNPs and haplotypes, and it is also observed that the alternative or mutant allele is associated with the more CN19 levels in the blood serum of people in compare with the people who have the wild type or wild type allele. Then we also perform the machine learning methods to detect the variables which have increase or decrease the risk of development of pancreatic cancer. So we use the logistic regression and classification regression tree analysis. Logistic regression is the statistical analysis method that constructs a statistical model to describe the relationship between binary and dichotomous outcome and a set of independent or explanatory variables. Classification regression typically helps to determine the particular data set and helps to create a model. So this is the logistic regression model, model one. In this, we check the risk prediction of clinical variable or demographic variable with the association of SNPs, and the gender, BMI, diabetes, and FOXQ1 SNP RS1124679 is significantly associated in the development of the pancreatic cancer. The second model predicts the demographic and clinical variables with the haplotype, and the red text also shows that the gender, BMI, and diabetes with the red highlighted haplotypes increase the risk of development of pancreatic cancer. Then a second machine learning model we use is the classification regression tree, and there we show that node one, if the person with these haplotypes have the more risk to develop the pancreatic cancer more develops in male, and if the person BMI is more than 24, he or she have the more risk to develop the pancreatic cancer, and if the BMI is less than 17, the person have lesser risk to develop the pancreatic cancer. Then we compare the allelic frequency in the southern Punjab, Pakistan population with the other with the frequency of other populations, and we show that the allelic frequency is almost near to the allelic frequency in the South Asian peoples of the other population. There is a conclusion of our research. We concluded that the BMI, smoking, diabetes, family history, SNP RS9.02.893 of FOXQ genes, and the showing haplotypes are contributed in the development of pancreatic cancer in southern Punjab Pakistani population. Limitation: we found that SNP RS1124679
वैसे
is not significantly associated with pancreatic cancer, while RS9502893 is associated with pancreatic cancer to a lesser extent. Maybe it is because we have a low sample size, and if the sample size increases in future, maybe that SNP also will be associated with the development of pancreatic cancer. The future prospects of our studies: the machine learning models, after counting various genetic and demographic factors, may help in early diagnosis of the disease or help in the predicting risk elevated pancreatic cancer. It will help in effective disease management and decision making medical practitioner and experts. These results will help in making personalized medicine. Thank you very much. If you have any question, you can contact me. Once again, thank you. Bismillahirrahmanirrahim. I am very thankful to the third International Brain and Biomedical Conference, which is organized by the Department of Physiology and Department of Pharmaceutics, Government College University, Islamabad, who have provided me a platform to present our work. Now, I am Dr. Azarfi, Associate Professor, Department of Zoology, Government College University, Islamabad. The title of my presentation is Epidemiology and Chemotherapeutic Assessment of Gastrointestinal Nematode Clumbalivia domestica from Faisalabad city. First of all, the NIFA project, helminths have been involved in causing infection, high mortality in domestic pigeons, considered as one of the critical weakness in the production of commercial pigeon in Pakistan. Pigeons have the heavy gastrointestinal helminthiasis that is characterized by emaciation, loss of appetite, anemia, paralysis, mucoid diarrhea. It also, there are different nematodes which are found in pigeons, that is dyspharynx species, roundworms, capillaria species, relatina species, which causes anemia, droopiness, emaciation, diarrhea, weight loss, etc. And also the reduce the egg production, also cause a chronic gastroenteritis, anorexia, and mortality in the of the pigeons. Pigeons fall into three groups. That is poultry pigeon, carrier pigeon, or racing pigeons. Lumbia livia, the scientific name for the common pigeon, is also the ancestor of the domestic pigeons. They have also their risk factor for the pigeons that are high environment temperature has been identified as a factor which alters the physiological parameter of birds. Most dominant factor for reduced productivity and overall health of the pigeon is the pathological legend caused due to helminth parasites. So these are the drugs which are used to treat the exotic birds, predominantly Ascardias and Caplaria species. The majority of these treatments have appeared to be safe and effective for the treatment of helminth infections. It is an anthelmintic compound used in the veterinary practice. Its main use for protecting against roundworms, strongyles, and pinworms, etc. Levamisole is also used as a dewormer. It's a medication used to treat the parasite worms. There are aims and objective to assess the nematode infestation in the domestic pigeon in Faisalabad, to collect and in vitro culture the eggs oocytes from cases of nematodes infestations, to assess the anthelmintic efficacy of drugs, albendazole, fenbendazole, and oxfenbendazole, etc. Then the materials and methods, the search project was carried out in Department of Zoology at Government College University, Faisalabad. In this investigation, prevalence and chemotherapeutic investigation of gastrointestinal and nematodes infestation have been analyzed. A total of 140 samples of pigeons of all ages sex were collected from the city of Faisalabad. After collection, all fecal samples were checked immediately or preserved in 10% formalin solution for future studies and transported to laboratory, Department of Zoology, Government College University, Faisalabad. Fecal examination are of two types, qualitative fecal examination and quantitative. In the qualitative examination, first of all, the two methods are adopted, direct microscopy and flotation method. First of all, the direct microscopy. A small amount of fecal sample was taken by toothpick and placed on a clean grease-free glass slide. The fecal sample was mixed thoroughly with 1 to 2 drops of water and debris were removed. A cover slip was placed carefully on the pieces to avoid bubble formation and slide was examined under microscope at 10x objective. The second method is flotation method. This technique was carried out for the detection of nematode eggs. Other method is the quantitative, that is called the McMaster techniques, egg counting techniques designed by specific for the helminths. The McMaster technique, samples of 2 grams of feces were weighed and mixed thoroughly in 20 ml of saturated salt solution. This was then sustained using sieve with an aperture width of approximately 1 mm and the debris discarded. Then 0.5 ml of the resultant solutes were transferred into a double chambered McMaster slide, mixing the solute again before piping into the second chamber. The slide was then left for 5 minutes to allow the fecal debris with a specific gravity higher than 1.2 to sink and helminth eggs. The chamber were examined microscopically using a compound microscope under 10 magnification. Chemicals and drugs. The anthelmintic drugs, ivermectin, oxfenbendazole, albendazole, and fenbendazole were purchased from the pharmacy. These are safe, inexpensive, potent, and effective and are broad-spectrum compounds. In vitro survival tests, anthelmintic drugs were prepared in increasing concentration of 0.51 to 510 and 20 milligram per ml in 0.9% neutral phosphate buffer saline supplemented with 1% dimethyl sulfoxide. In vitro condition was maintained at 37 degrees Celsius in a glass chamber biological incubator. Egg assay, fecal sample with the highest number of eggs per gram was selected. The eggs were repeatedly washed with distilled water before the experiment. Untreated eggs in water were used as negative control. The test was conducted in 5 ml test tubes. Approximately 9 to 110 eggs in 0.5 ml of water was placed in each test tube. Serial concentration of each medicine in total volume 0.5 ml in distilled water was added to make concentration 0.51 to 510 and 20 mg per ml. Medicines 1% was originally dissolved in dimethyl sulfoxide and distilled water. The test tubes were then covered and kept in incubator at 27 degrees Celsius. The experiment was conducted in duplicate for each concentration replicated 3 times. The larval development test, positive and negative control. Eggs were incubated at 27 degrees Celsius for 24 hours to obtain first stage larvae. The experiment was conducted in plastic cups of 20 ml according to the techniques described by Costa with little modification. Ivermectin 1% was used as positive control, while untreated eggs in water were used as a negative control. All samples were then incubated for 7 to 14 days at room temperature. The experiment was conducted in duplicates for each concentration and replicated 3 times. Then the statistical analysis, the prevalence of nematode species was statistically analyzed using student t-test. One way ANOVA was used to compare the efficacy of the three tests. Level of significance was considered as is lesser than 0.05. Then these are the results. First table shows the overall prevalence of gastrointestinal nematodes columbalia domestica. Five. There are number of species having the two genera was identified, that is Capillaria longicolis, Ascaridia gallei, Ascaridia columbi, Capillaria obsiginata, and Capillaria columbi, Capillaria pardinflata. This table shows the two species that are the Capillaria longicola and Ascaria Ascaridia galli are prevalence highly prevalent Ascaridia columbi. These species are highly prevalent. This is the pie graph of the overall prevalence of gastrointestinal nematodes of Columbaria domestica. This pie graph also shows the Columbia Ascaridia columbi and Capillaria species are the dominant species. This table shows the comparative enzymatic efficacy of three drugs. This table shows the LC50 and percentage of larval development of nematodes at different concentration of oxfendazole in pigeon. LC50 was the 4.966 milligram per ml. This table shows the LC50 and percentage of larval development of nematodes at different concentration of fenbendazole in Columbalia domestica, where the LC50 is equal to 4.789 milligram per ml. This table shows the LC50 and percentage of larval development of nematodes at different concentration of levamisole in pigeons, where the LC50 is equal to 5.102 milligram per ml. Treatment-wise incidence of gastrointestinal parasite. This table shows the different of drugs, enzymatic activity of the drugs, which shows that the levamisole has the highest enzymatic activity against the nematodes. Then the conclusion, the research findings indicated the presence of nematodes, including Capillaria longicolis, Ascaria galli, and Ascaria galli and Ascaria columbi, Capillaria obsignata, and Capillaria columbi, and Cardiflata, and in Columba livia domestica. The use of enzymatic drugs induced notable alteration in Columba livia domestica. Among the administered drugs, levamisole exhibited the most effective enzymatic activity for treating nematode infestation in domestic pigeons, surpassing the efficacy of oxfendazole and fenbendazole. Thank you. This is our presentation. Any questions, please?
Hello everyone. I'm very grateful to be here as a part of third International Brain and Biomedicines Conference 2026. A bit about me. My name is Asad Khan, and I'm affiliated with the Department of Biotechnology, Faculty of Sciences, International Islamic University, Islamabad, Pakistan. This project was carried out at the department under the supervision of Dr. Bashir Ahmed, who is associate professor. And this project is all about designing and development of molecular inhibitors of that protein known as papain-like proteases, which actually play a role in the viral application and in or escaping host immune response. Introduction and background. This virus, known as SARS-CoV-2 virus, that caused COVID-19, that took place back in 2019, that caused a major health issue globally, which the protein which actually plays an instrumental role in the viral application and preventing the host immune response is known as papain-like proteases. Because of his direct role in the viral propagation, this means this protein turns out to be a prime target for the therapeutic development of antiviral drugs. Although a lot of study and a lot of molecular inhibitors, synthetic molecular inhibitors have been developed, designed and developed against that protein, but still it faces issues like toxicity and low selectivity. And natural compounds, on the other hand, provide a diverse chemical space to be identified as a novel that protein inhibitors. And this is about these are strategies, these are ways that this virus can be blocked at multiple stages of its life cycle, such as blocking the spike protein from binding to the host human cells, such as human cells receptors such as ACE2, and its fusion with the membrane of that host cell. In this study, we executed some structure-based feature screening of natural and FDA-approved compounds using different databases, Campbell protein database, and there are various databases available where from which you can download your or you will pick up your ligand or that can specifically bind to your target protein or receptor. We in this study we try to identify some leading compounds who show the favorable binding and affinity and have strong molecular interactions such as heterogen bonding, van der Waals interactions. And on the next step, in the next step, we assess the drug likeness or drug related properties such as pharmacokinetics and the toxicity of these molecular hits, because it's a very important part and most important aspect of the drug development, because if we need to know how will that drug be working in the human body. Then lastly, we try to propose most potent and selective natural inhibitor based on binding affinity and other molecular characteristics to perform further molecular experimental validation. This is the layout, the flow chart of our methods. First of all, we retrieve our target protein from protein database using his ID number such as 6WX4. Plus, we also retrieve 100 plus Campbell ligands, and then we prepare the protein using UCFC chimera. This is also very important software where you can prepare your protein and you can remove your natural ligand which are already attached to the binding pocket of that protein, so we can design and check the docking of our own ligand or own drug to that protein receptor. We carried out vector screening, which is also very important, using empty open sequence of open web server. We have identified more than 1,000 FDA approved and more and natural compounds against that protein. After that, after that screening process, we carried out molecular docking using MOE software, that stands for molecular operating environment, using Umber 99 Phosphere. Then we validated our screening process, because it is very necessary to know that whether our screen compounds are inactive or active compounds against our target. Then the most important step, that is admit production, absorption, distribution, metabolism, excretion, and toxicity prediction of our drug and how it behaves in the human body using SysAdMe. So tools, online tool that is available, that is very frequently used tool and gives very prominent results based on different parameters. Then we identify lead compounds based on its binding energy. The more the active energy, the more is its binding energy to bind with these targets. Based on this molecular energy that is in kilojoules mole, we identified paraharcamide as our leading compound. This is our curve analysis. This actually validated that our screening process that we had carried out using MOE online servers to pick up our ligands and all that. And once you carry out this analysis, there when we go zero point 0.9 value, which is also an excellent performance, and let's give some binding affinity and classification for activeness, positive group which are actually active group against that our target, and negative group which are only 16%. So then we identified our leading compound, that is paraquamide, given with zinc ID, and showing that favorable binding energy of minus 9 kilocalorie per mole, and it interacted with the key active site residues such as glycine, serine, methionine, and compared to the other identified molecular hits, this compound showed favorable drug likeness properties as well, and most importantly, violate lipid sky violations, which is the most critical issues that occur with other leading compounds. That's why we picked that compound up for the further process. This is the 3D representation of the paraquamide that we code after molecular docking analysis, and we can visualize like through different softwares. But we use Discovery Studio. You can also use by mole, but in our case we use Discovery Studio. This is the three interaction of our protein and ligand with this polar charge nesting and basic amino acids, and these are the properties that with analysis like absorption, like solubility, skin permeability, blood-brain barrier permeability, and high absorptions, and most importantly, it cannot interact with other drug, minimizing the risk of drug and drug interactions, which means it can work individually. These are some physiochemical properties of dilating compound with molecular weight, chemical formula, number of hydrogen donor acceptors, depends violation, and GI absorption. This is very crucial parameters and how it will behave, how it will act in human body after its final trial. So concluding that we identified pyraquamide as our stronger and selective molecule inhibitor candidate because it showed high binding affinity, violated zero violations of Lipinskaya rules, and good pharmacokinetic properties. And the is to further work further work will be required to conduct in vivo and clinical testing for the confirmation for its full drug development. These are some references, and thank you very much for that opportunity.
Bismillahirrahmanirrahim. Assalamu alaikum everyone. I hope you are all doing well. First of all, I would like to introduce myself. My name is Muhammad Zishan Jamil and I am from the Department of Zoology, Radiant College, Nankana Sahib, affiliated with Government College University, Faisalabad. It is truly honor for me to present today on an important topic related to modern biology and environmental science. As we all know, climate change is rapidly transforming our world and creating serious challenges for the survival of many species. Today, I will be discussing how epigenetics plays a crucial role in helping organisms adapt to these environmental changes. My topic is the role of epigenetics in species adaptation to climate change. Adapting to a warming world. Environmental pressures. Climate change is rapidly altering ecosystems, exerting strong pressure on survival. Traditional genetic evolution via mutation is often too slow to keep pace with these shifts. Epigenetics enables rapid and reversible molecular responses to environmental stress. Defining epigenetics. Gene expression, heritable genes, expression that do not involve alterations to the underlying DNA sequence. Environmental bridge act as a link between the environment and the genome, controlling gene activity through molecular switches. Molecular mechanisms. DNA methylation, adding methyl groups to DNA to silence specific genes. Histone mode, altering chromatin structure to control gene accessibility. Non-coding RNA, regulating gene expression at the post-transcriptional level. Environmental stressors, rising temperatures, extreme drought, ocean acidity, ultraviolet radiation. These factors trigger epigenetics modification to maintain biological homeostasis and survival. Phenotypic plasticity, one genotype, many traits. Plasticity is the ability of an organism to change its phenotype in response to environmental stimuli without genetic changes. Epigenetics serve as the molecular foundation for this plasticity, allowing organisms to adjust traits like metabolism and behavior. DNA methylation, the silencing signal. Addition of methyl group to DNA, typically at cytosine phosphate group, this modification usually suppresses gene expression. Case in point, methylation patterns change rapidly to regulate stress response genes during the climate shifts. Histone modification. Chromatin architecture modifying the tails of histone proteins alters how tightly DNA is wrapped. Acetylation typically activates genes while deacetylation represses them. This allows for rapid gene switching in response to climate signals. Transgenerational inheritance. Inheritance epigenetic traits are passed to offspring without DNA change. Parental stress. Parents exposed to climate stressors develop epigenetic marks. Readaptation. Offspring may be born better, adapted to same stressors. Marks that escape resetting provide a multi-generational survival advantage. Case study, animal resilience. Fish rapidly adjusting metabolic rates to rising temperature. Marine biota, epigenetics, regulation against ocean acidification stress. Insects, altering development and diapause timing. Case study, plant adaptability, immobile survivors. Plants rely heavily on epigenetics because they cannot move away from stress. Modification, facilitate drought resistance via gene regulation. Improve water use efficiency, flowering time adjustment. Advantages of epigenetic adaptation, rapid response can occur within a single generation, unlike mutation. Reversibility, organisms can undo marks if conditions stabilize. Genome stability, no permanent sequence mutation required. Sudden stress, crucial for coping with rapid environmental fluctuations. Limitations and gaps, stability can be transient or reversible, permanent across all generations. Mechanism, molecular switches, methylation, DNA sequence mutation. Speed, rapid, single generation, slow, thousands of years. Future outlook, limited field studies available, well-documented evolutionary path. Conclusion, epigenetics provides the short-term molecular flexibility for immediate survival, while genetic evolution ensures the long-term persistence of species. Synthesis of adaptation theory. Understanding this molecular bridge is vital for future conservation strategies and ensuring resilience in the face of global climate change. Thank you for your kind attention.
Hello, hi, assalamu alaikum. My name is Waqas Ashraf. This presentation is recorded to the third International Brain and Biomedicine Conference 2026. My topic is genome annotation and in silico characterization of biocontrol related genes in indigenous business strain. These are my outlines. Introduction. Soil-borne fungal pathogens like Physidium, Rhizoctonia, and Alternaria cause 20 to 40% annual crop losses worldwide. Chemical fungicides are effective but raise environmental and resistant concerns. PGPR offer eco-friendly disease management. Bacillus species are particularly promising due to spore-forming ability and production of antifungal metabolites. Strain TD11. Indigenous Bacillus species TD11 isolated from the local rhizospheric soil of Dera Ismail Khan, UP, Pakistan. Previously reported by Malik et al. 2022 to suppress tomato fungal pathogens via catalytic activity. Rationale of the study. Information on the genome of an isolated strain of Bacillus from local regions remain limited. Strains are isolated from other regions are model strain often show difficulty in adapting to local soil conditions. This study examined an indigenous predicative strain of Bacillus isolated from the local environment using computation methods and approaches for analyzing the genome. This analysis identifies genes, proteins, and nucleotide sequences that are used as biocontrol against fungi. Objectives: to assemble and annotate the draft genome of an indigenous Bacillus strain with putative biocontrol activity, biocontrol potential. Number two, to identify and characterize biocontrol associated genes and BCGs using advanced in silico computational tools. Methodology. This is the pipeline. First of all, we use Windows 11 and we set up the Linux for Windows 11. Then we retrieve our query genome and reference genome from GenBank. Then we check the quality of our genome by fast QC and trim the low reads by trigonometric and reassemble the genome by speeds and annotate our genome for CDS and tRNAs by PROCA and circular visualization by proxy and smash and BCG profit. Identify different BCGs. Multi-gene blast. Compare the all genome and big fem analysis. Show the distance of our query genome from other Bacillus genus genome. Deep CMS used for the prediction of structure and function of the novel genome, novel clades, and novel nodes. CSZY database show the biocontrol enzymes. MEFT by MEFT we use for alignment. Fast ANI show the average nucleotide identity. And last but not least, Mega we built a tree by Mega. Target organism and genome retrieval. We target the indigenous Bacillus strain isolated from the local environment and previously identified by Malik et al. For tomato fungal pathogens. This project was funded by the Genome Project. The excision number for our query sequence is this. Use single and elemental reads and fast QM file. Comparative analysis and tree building. For comparative analysis, we retrieve different Bacillus strains. This is our query sequence, query genome, and these are the reference genome retrieved from NCBI. And this is the outgroup genome. These are all the name of the strains, and these are all the excision number of every particular genome. Results. Quality control and reprocessing. When we apply the fast QC, high quality data were obtained, approximately 2.95 million reads. Every read has 36 to 251 base pair. GC content is 34%, which is constant all over the reads. Strong score means Q30 plus across all reads. Clean library, no adapter contamination found. Automatically keep 100% of the reads. This is the table and this is the graph, which is produced by created by the fast QC and Chemometric. We reassemble our genome after the trigonometric. We found 21 to 127 kmers. We found our genome complete from 21 to 127 kmers. We found our genome complete. We annotate our genome by PROCA. We found 6.25 million base pair the length of our genome and 96 condex, 600, 184 CDS, 104 tRNAs, 14 rRNAs, and 1 tmRNA. This is the circular map of our genome created by proxy. We can saw that uniform CDS distribution. Biosynthetic gene cluster analysis using anti-smash identified means 18 BCGs across 13 contexts, covering 9 BCGs classes, including
Naveed,
NRPS, PKS, IPPS, trypin, and SAT4, etc. BCG profit analysis, we found another 284 BCGs for trypin and 100 fold, which is already mentioned and smash analysis. Discovery of novel BCGs. Multi-gene blast analysis. We compare our query sequence, query genome with 7 reference Bacillus genome. Several clusters show homology with Bacillus cereus and Bacillus subtilis, but these 3 clusters show no significant homology with any other reference genome. Node 1, node 30, and node 38. Now we use big fam analysis and check the distance of the 3 nodes, which is the good, and these are all no similarity with Bacillus, but some similarity with some other bacteria. Predictions, structure prediction of novel nodes using deep CMS AI2. This is the node 1, which is similar with polyene, pyrimidine, dione derivative. Node 30, which is the similar with staclysine C3, and node 38, which is similar with tyrosine. Function of node 1. Mostly, function of node 1 may be enzyme inhibition, membrane encryption, possibly antifungal and antibacterial activity.
ਅੱਛਾ
Likely function of node 1
ਇਨਰ
is
ਕਿਤੇ ਪਿਆ ਵਾ
antimicrobial
ਕੋਈ
or enzyme
ਉਤਰਿਆ
inhibitor.
ਹੋਇਆ
Antimicrobial or enzyme function of node 30. Likely function of node 30 is antimicrobial peptide because the molecular weight is very high. Sulfur suggests membrane destruction, pore formation, and bacteriocytic activity. Function prediction of node 38. Likely function of node 38 is broad spectrum antibacterial peptide because broad spectrum antibacterial activity is dependent of cell membrane integrity, use and topical antibiotics.
ਇਹਨਾਂ ਇਹ ਪੁਰਾਣੇ ਕੋਈ ਉਤਰੇ
This is the analysis
ਜਾਂ
of
ਇਹਨੂੰ
every
ਅੱਛੇ ਤਰੀਕੇ ਨਾਲ ਚੈੱਕ ਕਰ ਲੈਣਾ ਇਨਸਾਨ
we find different genes already published in Hammer, Diamond, and DBCAN database. These are all the biocontrol genes present in our genome, like chitinase, cellulase, glucokinase, protease, and these are the gene ID present in our genome, and this is the function of
ਅੱਛਾ
every particular gene.
ਇਹਦੇ ਵਿੱਚ ਮੇਰਾ ਖਿਆਲ ਗੈਸ ਗੂਸ ਦਾ ਕੋਈ ਇਸ਼ੂ ਆ
Fast ANI taxonomic placement. This is our query genome, and these are the reference genome. So our query genome is closely related with the Bacillus cereus, which is
ਚਲੋ ਠੀਕ ਹੈ
98.41% similarity, and the phylogenetic tree of
ਉਹ
16
ਇੱਧਰ
RNA
ਆਏ
shows that Bacillus species TD11 is closely related with the Bacillus cereus.
ਚਲੋ ਨਹੀਂ ਆ ਕਰ ਦਿਓ ਫਿਰ
So this is the conclusion of our entire presentation. We found bacteria and rhizosphere of the soil. The total length of our genome is 6.25 million base pair, and all these are information are here. This is the acknowledgement because we have less time and didn't discuss these acknowledgement and conclusion. This is the differences. Thank you.
My name is Sarita Kenya, and it's not undergraduate study where I did for the completion of my bachelor's degree in physiology and did mentorship and the supervision of Dr. Saravanan and Dr. Kamika. My study cited behavioral modulation of sleep, the role of sleep hygiene in insomnia management, and psychophysiological evaluation in younger. So as we all know, let's skip to the fundamental biological aspects, and it plays a very critical role in maintaining our physical health, our emotional stability, and also our optimal cognitive functioning. And despite its importance, poor sleep prevalent among young adults. It could be due to the academic pressure. It could be due to their irregular sleep schedule. It could be due to their increased sleep exposure. And if these issues persist over time, they may also contribute not only to the chronic sleep disturbances, but also to the broader health consequences like impaired cognitive performances, mood disorders, anxiety, depression, and long-term cardiometabolic. Since any of these contributing factors are behavioral in nature, they can potentially be modified. And this makes sleep hygiene a practical and non-pharmacological intervention for improving sleep outcomes. The aim of this study was to evaluate whether structured sleep hygiene interventions could improve sleep quality and reduce insomnia severity. So for this very reason, I employed a randomized controlled design for this study, and the participants that were taken were aged 18 to 25 years old, and they were recruited and randomly assigned into two groups. So there was a control group and there was an intervention group. Control group had to continue their usual daily routine without any kind of behavioral modification, but the intervention group had to follow a structured sleep hygiene program for 4 weeks. So, in this sleep hygiene program was designed, particularly designed to target modifiable behavioral habits that influence sleep, that influences sleep. So, like, they had to maintain a consistent sleep and daytime every day, which included their weekends too. Then they had to avoid their screen exposure, especially in the evening, to reduce blue light stimulation. They had to limit their caffeine intake, and before sleeping, they had to optimize their sleeping environment. Following the relaxing pre-sleep routine, it could be breathing, it could be light stretching, or any kind of calming activity which could promote their sleep readiness. Then the sleep outcomes were assessed using both subjective and objective measures to ensure evaluation. So for subjective measures, we used Pittsburgh Sleep Quality Index to assess their overall sleep quality. We used Sleep Hygiene Index to evaluate their behavioral sleep efficacy. We used insomnia severity to measure the severity of insomnia symptoms. And for the objective assessment, sleep data was collected using a mobile application, which was Sleep Cycle mobile application. It provided the information on their sleep latency and their sleep patterns. The statistical analysis included ANCOVA for the subjective outcome and the linear mixed model regression for their objective outcome. The result demonstrated significant improvement in the intervention group compared to the control group. Like for sleep quality, the PSPI scores in the intervention group decreased from 11.3 to 7.9, and it indicated a meaningful improvement in risky sleep quality. In contrast, the control group showed no significant change. Similarly, the insomnia severity also showed a strong improvement. Like for example, ISI score reduced from 18.8 to 5.1 in the intervention group. It suggested a substantial reduction in the insomnia symptoms. Then for the objective sleep data, there was a significant reduction in their sleep latency, which meant that participants were able to fall asleep more quickly after implementing the sleep hygiene practices they were given to follow. So, in conclusion, the study demonstrated that a structured 4 week sleep hygiene intervention is very effective in improving sleep quality and reducing insomnia severity among young adults. And the findings also highlighted that simple behavioral modification like regulating your sleep schedule, reducing your screen exposures, and reducing your bedtime can have a meaningful impact on your sleep health. Therefore, sleep hygiene represents a low-cost, accessible, non-pharmacological approach for managing sleep problems in young adults. Thank you for your attention. I would be happy to take any questions. Thank you.
Assalamu alaikum everyone. I hope you all are doing well. My name is Adina Ijaz and I will be presenting on behalf of my group. My group members are Barisha, Iman, Laiba, Rabia. We are final year students of Bachelor of Eastern Medicine and Surgery program at Hamdard University main campus Karachi from Faculty of Eastern Medicine. This research was conducted under the supervision of Hakeem Muhammad Anas Shamsi and Dr. Hakeem Sayyid Zahurul Hassan Zaidi. Today I will be presenting our topic on cognitive decline and its association with temperament among youth. Before moving forward, I would like to examine some important terms of our field. Firstly, temperament. Now you must be wondering what is temperament. Temperament refers to the natural balance, that is homeostasis of person's body and mind. In simple, it is the individual's overall nature and how their body reacts to different conditions. According to Unani medicine, every person has a temperament based on four humors, that is sanguineous, phlegmatic, bilious, atrabilious. And every human has their own qualities and characteristics. And all these qualities are reflected in person's personality. At this point, you might be asking about humors. A humor is one of four natural body fluids. When these humors are in balance, the body remains healthy, but when there is imbalance, either increase, decrease, or change due to any environmental or lifestyle factors, it can lead to dysplasia or maltemperament. Maltemperament can be managed by correcting the imbalanced humor through the six essential factors. That is environment, diet and drinks, movement and rest of body, movement and rest of psyche, sleep and wakefulness, retention and excretion. Now, moving to forward to the objectives of study. First one is to assess the level of cognitive decline among youth. Secondly, to assess cognitive decline in youth and its relationship with temperament and maltemperament. To achieve this, we conducted a cross-sectional study on 130 participants aged 15 to 24 years. Cognitive function assessed using cognitive failure questionnaire developed by Broadband and temperament was evaluated as described by Ibn Sina in his book The Canon of Medicine. Now let's move towards the graph. I will briefly explain what the pie chart shows. It shows four scores, 1, 2, 3, and 4. 43% of people scored a 1, that's the largest group. 31.5% scored a 2, the second largest. 21.5% scored a 3, and then only 4% scored a 4. Together these add to 100%, so this chart includes everyone. In short, most people got a 1 and a very few got a 4. Now the first paragraph shows if mild temperament affects cognitive score, most people with phlegmatic temperament scored low or average. Sanguineous and bilious almost all scored low. Atrabilious was spread out. The p-value is 0.031, which means this pattern is likely real, not by chance. Now we look at whether temperament relates to cognitive scores in 130 people. The second paragraph shows some difference, but the statistical test gave p-value of 0.37 and 0.30, both well above 0.05. That means no significant relationship and pattern we see in is likely judged by chance. These findings are further supported by graphical representation generated using SPSS version 26. Overall, these graphs clearly support our finding that temperament, especially phlegmatic, plays a significant role in cognitive decline. Phlegmatic individuals are generally slow in response, calm, and may have reduced mental alertness. Now, what are the impact of our study? Individuals with a phlegmatic temperament can be identified earlier and provided with targeted preventive measures by addressing modifiable risk factors. Since cognitive decline is more associated with phlegmatic humor, management will be through bilious temperament or pharmacotherapy. This also supports the use of personalized medicine to improve outcomes. Thank you for your attention. I am honored to have this opportunity to present our work. Detailed references are available on this poster.
Everyone, my name is Ariba Muhammad Ali, and today I'm going to present my undergraduate research, which I did for the completion of my BS degree program under the supervision of my teachers, Dr. Sadaf Ahmed and Dr. Islam Mirza. So the title of my research is Non-Pharmacological Ordinary Intervention for Sleep, Psychophysiological Comparison in Shift Workers. So let's talk about the background of my study. Night shift is very common in people in our society. For example, if we talk about our health society, doctors, and not only which normally controls the sleep cycle. So due to this disturbance, our sleep difficulty occurs, and sleep duration becomes less, and our mind becomes exhausted, and our sleep quality also becomes poor. And eventually, these problems causes our cognitive function. It affects our cognitive function as well as our attention, memory, and physical and mental health. It all affects these points. So the researchers mainly focuses on non-pharmacological interventions. These are safe, non-invasive, and low cost, and they don't have any kind of side effects. In the supported method, audio listening, in which we listen to nature sound, and it relaxes our body and it makes our sleep better. And it overall relaxes the body. So this relaxes the body and helps to get a better sleep. Okay, so after that, discuss the objective. So was evaluate how nature sounds affect the sleep quality of night shift workers. Do they really have effect on their sleep quality? Like do they improve their sleep quality or not? So was the let's talk methodology. My response it was randomized control trial, and total there were 30 participants, and with 30 participants chose night shift workers for this. And my research was totally of 2 weeks duration, and the people were under 20 to 40 years old. The night shift workers we chose for 20 to 40 years. Okay, so now the participants were randomly divided in groups. Number was the intervention group, and other was the control group. In intervention group, 15 participants were included and listened to music for 20 minutes. Like before their sleep was, so they used to listen to the nature sound which we provided them. Nature because we were a lot of sound. Like these sounds calms the body, calms our mind. And there are a lot of music like classical music, but we chose nature sound as an intervention for the participants. And the other group was control group. Control group did not receive any kind of intervention. We just let them to fill our forms before and after the intervention period, like before 2 weeks and after 2 weeks. And in intervention group, participants listened to nature sound for 20 minutes, and we provided them nature sounds. Now let's talk about their sleep quality. So their quality using was the peak sleep quality index and MQS scale subjective sleep. The measurement became 2 weeks after 2 weeks duration of both groups, intervention group and the control group. And these are questionnaires. These are basically questionnaires which have many kinds of questions according to the sleep. So we use them for their assessment. We also used participants. We also provided them post-intervention form in which we got their we got to know about their experiences that they experienced during the 2 week intervention, and then they fill out according to that. So now I would like to talk about the results. So the intervention group showed significant improvement in their sleep quality. For example, the PSQI scores decreased from 8.13 at baseline to 5.87 after the intervention period. And this indicates the better sleep quality, and it also indicates that they had showed some improvements in their sleep quality. And if we talk about the control group, so there were not any significant changes, any like minimal changes were due to some changes in their normal routine as they did not get any intervention. So the results of my research study suggests that nature sounds reduce the stress of our participants, and it promoted the better sleep quality. It was the better observed with them. And when we asked them to fill the intervention, so showed their positive comments to the intervention, which we gave them. And in the SQI sleep quality index scale, so not any significant changes were observed. So now I would research that show effects on the night shift workers, and they are simple, low-cost, non-invasive method for improving the sleep quality of the night shift workers. So they can use this intervention in their daily routine, as it has not any kind of side effect because it is non-pharmacological intervention. So this does not have any kind of side effect. So that's it, and it was about my research. So thank you everyone.
Assalamualaikum everyone. My name is Mariam Jan, and I'm presenting my MPhil research work from Neuroscience Research Lab, Biochemistry Department, University of Karachi. With in start, I will just give you a quick background of my research. So, amphetamine is the FDA approved drug which is used for the epilepsy and ADHD. This is the most commonly misused drugs, which leads to the anxiety and dependency like symptoms, and it arises a major public health concerns. So, amphetamine basically affects by targeting the dopaminergic system in the brain, but it has been less commonly has been discussed that it also affects by targeting the gut-brain axis. I give you a little background about it. So basically, chronic amphetamine do the induce the gut dysbiosis, which is just disrupting the balance between the good gut microbiota and the bad gut microbiota, which leads to the gut inflammations, leaky gut. That leads to the peripheral inflammations, leaky blood-brain barrier, and ultimately leads to the neuroinflammations and causing the anxiety-like symptoms and dependencies. So here, this gives us the foundation for our research question. Can we attenuate these symptoms, which is leads by the amphetamine misused? Can we attenuate this by giving the probiotics? Or the objective of the study is to evaluate whether a multi-strain probiotic EcoTac could attenuate behavioral biomarkers of amphetamine dependence in the male Western rats. So I just give you the methodology about it. I take the male Western rats divided into the four groups. One group is water groups, another is second probiotic groups, amphetamine groups, and the amphetamine probiotics groups. We both given consecutively at the same time. So probiotic dose are given is an exponential line CFQ per day. Amphetamine is given for 2.5 mg per day, and the water 1 ml. We give this doses for like 2 weeks, and after that we sacrifice animals and we get the neurochemical profile analysis in which we analyze the serotonin level, dopamine level in the prefrontal cortex, codates, nucleus accumbens region, which is leads through which is analyzed by HPLC techniques, and which we do the antioxidant profile analysis through lipid oxidation and acetylcholine enzyme, antioxidant enzyme, which is again GSH, and the catalyze enzyme. We check their levels in brain and also in plasma through their biochemical assays. So I share you our key findings of research. So we check the effects of probiotic on antioxidant profile in amphetamine-induced anxiety and dependencies. So you can see in my graph there is a upregulation of lipid or oxidation product, which shows there is an increase in oxidative stress when amphetamine given only. When the probiotic is given, it's just down-regulate that oxidative stress. And for the acetylcholine esterase, you can check that it's down-regulate the acetylcholine esterase enzyme in the brain, in the plasma, which show there is an upregulation of acetylcholine level, which show that there is some kind of dependency like symptoms arising. Say, and probiotic just upregulate this acetylcholine esterase enzyme activity, which show that it's just attenuate these symptoms. And same goes for the SODs, the catalase glutathione enzyme. You can say these three are the antioxidant enzyme, which is very beneficial for us. It just down-regulate these three enzymes, increasing the oxidative stress, while just the probiotic given can act by upregulating these antioxidant enzyme. And in effects of the probiotics on neurochemical profile and amphetamine-induced anxiety and symptoms, we check in street 4H nucleus. So you can see there is an upregulation of dopaminergic level, which shows that there is which is related to the dependency like symptom. There is an upregulation of dopamine, but probiotic just attenuate the dependency symptoms by down-regulating the dopamine levels in these areas, brain region area. And you can see that serotonin level, this down regulation of serotonin level in that areas brain getting because it has basically been proved to numerous researchers that down regulation of serotonin level in the brain is related to the anxiety and it just upregulate in the probiotics potency. So summarize these results support the gut-brain axis as a promising therapeutic target, highlights probiotic as a novel dopaminergic protein strategist for amphetamine dependency. So there's two interesting things about my research. One is it's a novel non-dopaminergic therapeutic strategies, and second, it's a probiotic, so there is no side effects like other. This work as part of my research contribute to the growing evidence what happened in the gut does not stay in the gut. It shapes the brain, its behavior, and ultimately contributions to the addiction. If you like take a full copy of this poster, you can scan this QR code, and if you would like to directly contact to me like to just email on that. I would like any question if you have. Thank you.
Last presentation guidance was to ask last presentation guidance was to ask program.
So, dear participants, now we are closing to the session. Hopefully, you all have enjoyed all the presentations. We are full of knowledge and indicating the modern research trends. So, we are hoping that you will be available tomorrow and we will join according to our schedule in the morning. Thank you very much for your presence, listen, and listening and interacting with the participants. Allah Hafiz.
हो रहा होता है ना? इस नए और अहम जायजे में खुशामदीद। चलिए सीधा IBBC 2026 यानी थर्ड इंटरनेशनल ब्रेन एंड बायोमेडिसिन कॉन्फ्रेंस की पीयर रिव्यूड तहकीक और इसकी जबरदस्त तिब्बी जिद्दत की 26 की दुनिया में हमेशा कुछ ना कुछ नया और हैरानकुन हो रहा होता है ना? इस नए और अहम जायजे में खुशामदीद। चलिए सीधा IBBC 2026 यानी थर्ड इंटरनेशनल ब्रेन एंड बायोमेडिसिन कॉन्फ्रेंस की पीयर रिव्यूड तहकीक और इसकी जबरदस्त तिब्बी जिद्दत की तरफ चलते हैं। यहाँ न्यूरोसाइंस और फिजियोलॉजी का एक ऐसा कमाल का मिलाप देखने को मिल रहा है जिसने साइंसी तहकीक की सिमत ही बदल दी है।
खैर सीधा इसके मेन थीम पर आते हैं। इस कॉन्फ्रेंस का बुनियादी और मुस्तकबिल पर नजर रखने वाला मकसद बड़ा दिलचस्प है। जहनी सेहत, जिस्म और माशरे के आपस में जुड़े गहरे ताल्लुक को आर्टिफिशियल इंटेलिजेंस के इस नए दौर में समझना। और यकीन मानिए, ये थीम कोई आम सी बात नहीं, बल्कि ये आने वाले वक्त की पूरी मेडिकल डायरेक्शन तय करने वाली है। अब एक बहुत ही अहम सवाल जहन में उभरता है, वो ये कि AI और नैनो टेक्नोलॉजी हमारी रिवायती न्यूरोसाइंस के साथ मिलकर मेडिकल इंटरवेंशंस में आखिर किस तरह इनकिलाब बर्पा कर रहे हैं। दरअसल क्लिनिकल रिसर्च और मुख्तलिफ फिजियोलॉजिकल स्टडीज़ का यही वो टर्निंग पॉइंट है जो मौजूदा तहकीक को बिल्कुल एक नए और एडवांस्ड रुख पर ले जा रहा है।
इस एक्सप्लेनर के सफर में हम AI पर मबनी बायोमेडिसिन के दौर से शुरुआत करेंगे, फिर न्यूरोसाइंस के नए रुझानात, अप्लाइड फिजियोलॉजी में आने वाली जिद्दत, न्यूरो और फिजियो
हाँ जी, अस्सलाम वालेकुम। हाँ ठीक है, इसको अब क्लोज कर दो। हम्म। अच्छा अच्छा। चलो।